Good afternoon all together and a warm welcome to our swine feeding webinar regarding nitrogen and protein reduction without losing any performance or profitability. So my name is Ursula Bläser and I'm the sales manager at Jefo, responsible for Germany. And I will guide you through the webinar, which is going to start with a short introduction of Walter and Anaïs themselves. And then I'm going to give you a short presentation about Jefo and what we are doing at Jefo. And after that, we are going to start immediately with a presentation of Walter, which is divided into 3 pitches. And after each pitch, you will have the possibility to ask questions. Please be so kind and put your questions in the question box, and we are going to ask this question in the discussion round. And every question will be answered. If it is not during the time of the discussion, we are going to answer every question after the webinar in mailing form. So, and after the presentation of Walter, we are going then over to Anaïs Augé, my dear colleague from Jefo, and she is going to present you the solutions Jefo can give to you. So now it is time to start with a short introduction round, and I'm starting with Professor Walter Gerrits from University Wageningen, where we are very happy that he found the time to support us in this webinar. And so, Walter, the word is yours.
Yeah, thank you, Ursula. So my name is Walter Gerrits. I work as a professor at Wageningen University in the Netherlands at the Animal and Nutrition Group. I'm very pleased that I can give a presentation about something that is very close to me, low protein diets and pigs. I think it's a very timely topic for this webinar. My area of expertise is mostly on digestive physiology and metabolism in different species, but mainly in pigs.
Thank you very much. Anaïs, would you also like to say something already about yourself?
Yes, sure. Thank you, Ursula. So good afternoon, everybody. So my name is Anaïs Augé. I work at Jefo since 1.5 years now as a swine and aquaculture market manager.
Thank you very much for this short introduction. And then I'm starting now with showing you a little bit about who Jefo is and what Jefo does. I hope everybody can see my screen. So what you can see here is the company, and Jefo is a family-owned company in the eastern part of Canada, quite close to the city Montreal. So this is Quebec, the French-speaking part of Canada. And Jefo was founded in 1982 by the animal scientist Jean Fontaine, and it is now already in the second, or let's say the second generation is now already in leading positions of the family. So this means it is still a family-owned company. We are active now in more than 15 countries all over the world, and of which Europe and the North African states are managed by the European Center, located quite close to the very beautiful city Nantes. And the vision of Jean Fontaine when founding this company has been to create a healthier circle of life on a more sustainable planet, which means that his mission is to imagine, develop, and also share the next era of precision nutrition. And this has led to the company's commitment, Life Made Easier, which means that the life will be made easier for the animals, for the nutritionists, for the veterinarians, for everybody who is doing around animal housing, animal nutrition, by having simple solution, and it's in his hands to fulfill the vision. All products that Jefo has developed or is developing are tested in at first at test stations, and we are very proud that we do have own test stations for poultry and also for swine. And regarding swine, we in the research center we have all pigs are of the same maternity origin, and we can do trials in weaning, growing, and finishing pigs with 32 pens of 12 weaning piglets and 52 pens of 8 pigs for growing and finishing periods. So our, let's say, we are one of the world's leading companies when it comes to protected products. We are using the specific Jefo matrix technology. And also we are a world leader in the production of enzymes and especially of fermentation extracts, which we also want to talk a little bit about today. And with this, we have a wide range of products available for nearly every species, which gives us the possibility to make life easier to everybody. But now I would like to give the word to Walter to start his presentation. Thank you very much.
Thank you, Ursula. Let's share my screen now. Can everybody see my screen now? Yes. Okay, very pleased to talk about the low protein diets in pigs today. First of all, I would like to acknowledge my co-author here on this presentation, Ilaria Menusi. She is a PhD student in our group, and she's been very helpful in compiling this this presentation. Typically, what we do is we'd like to show, let's say, pretty pictures. And here, this is not a very nice picture to look at. From a marketing perspective, it may not be a very wise thing to do, but I hope by the end of my presentation, you understand why I show this picture. I will present 3 pitches to you this afternoon. And first, I will focus on the potential of low protein diets to reduce emissions. The second pitch will be about the effects of low protein diets on animal behavior. And the third pitch will be on feeding low protein diets under low sanitary conditions. And I would like to emphasize that I will have a short break between each of the 3 pitches to allow questions and discussion. About low protein diets, and maybe you have seen this report of the European Commission being published in 2017. It's a very thick report with a lot of information there. But typically what I learned from it is that they say that low protein diets and phase feeding, they are the most effective means in reduction of ammonia emissions while maintaining similar performance. And they have computed in this report that per percentage point reduction of dietary crude protein, the ammonia emissions could be dropped by some 10%. And this is I think that this is valid within the range of, let's say, 17 to 12% of crude protein. And often what you see is simultaneously with a reduction in dietary protein, you see a drop in the pH of the slurry and an increase in the dry matter percentage. And these two effects will also help to reduce emissions. Also, what the report states, and I put it within quotes, is that there are no health and welfare implications as long as requirements for essential amino acids are met. And I don't disagree with this, but I think it also implies that we really need to make sure that we do know the requirements for essential amino acids. And there may be requirements that are different under different circumstances. I do not only want to show you the cover of a report. I think this research by Zhao et al. Published in 2019 is a very nice example of the many studies that are out there in literature that show the effectiveness of reducing dietary crude protein. This is a study, relatively small scale, with 18 pigs on 3 different diets, a high, a medium, and a low protein diets in four-phase feeding. The dietary crude protein was reduced in the high protein treatment from 17 to 16, 14, and 13%, and the medium crude protein treatment was 2 percentage points lower, and the low crude protein treatment was 4 percentage points lower than the high. And what you typically then see in many studies, and this one you see on the horizontal axis, the different weight ranges, and on the y-axis you see nitrogen retention. Typically what you see here is very little non-significant effects on nitrogen retention, although these may be significant when you blow up the numbers of pigs. Here there is a reduction here, but for the rest it's fairly similar. So you can do this while maintaining, let's say, similar performance. But when you look at the losses of nitrogen through feces plus urine, here depicted on the y-axis, you can see that it really decreases in all weight ranges significantly and also quite substantially. And they have also been measuring the ammonia emissions from the slurry after 4 hours after collecting. And what you see here also is a quite strong reduction in every weight category. You may wonder why this effect is, whether this effect is linear or not, because it seems to be that from here, from decreasing from medium to low, the effect may be smaller than from high to medium. And this may be related to the effects not being really linear in the low range. So if you go down from 30% back to the low protein diet, actually it means that you're up to 9% of crude protein here. But effectively what you do if you reduce the dietary crude protein concentration is that you reduce the quantity of imbalanced amino acids. And some of them may be essential amino acids and some of them may be non-essential amino acids. I think the major portion of the nitrogen that you save in excretion is originating from a reduced imbalanced non-essential amino acid supply. And the general dogma is that you can synthesize non-essential amino acids from nitrogen and the carbon source. And this is nicely illustrated by the research in Canada from Mansilla. And what they did here is they had 3 dietary treatments: a control, a low urea infusion, and a higher urea infusion treatment. And the urea was infused into the cecum here. And they depicted nitrogen retained on the y-axis. And what you see, and I have to say here that these diets were deficient in non-essential amino acids. When under these conditions you supply nitrogen in the form of infused urea, you can see a clear effect on retention of nitrogen. And if you look into the efficiency with which the urea nitrogen is incorporated into body proteins, you can see it's fairly complete. In other words, when non-essential amino acids are limiting, pigs are capable of using nitrogen from urea. And I think it's also nice to see that they've tried also in different studies to feed ammonia, and they have also been successful in doing so. So you can reduce dietary crude protein, but you have to make sure that you supply a nitrogen source, and ammonia or urea could be a good nitrogen source to do this. But it's not true that the sky is the limit, because you may indeed run into problems with non-essential amino acids. And this is a study from Silva et al. In Canada also. They used 96 weanling piglets, very young piglets, and they had 3 treatments. A control diet, in which they in a couple of weeks reduced crude protein from 24 to 20.5%, and 2 low protein treatments, either supplemented with glutamine or supplemented with glycine plus serine. And the low protein diets were really low protein for these young piglets. Crude protein was reduced from 20.5 to, let's say, 15% of crude protein. And they used skin collagen as an indicator of skin collagen abundance as an indicator of potential glycine deficiency, because glycine is roughly 1/3 of the protein in skin collagen. And what they did during 2 phases of their study, they compared the control versus the glycine plus serine supplementation and the glutamine supplementation. And what they saw is that in the low protein diets, you see a clear reduction in skin collagen abundance in the treatment that was only supplemented with glutamine, but this was compensated for when you supplement the diets with glycine plus serine, and the same in phase 3. And apparently they found that skin collagen abundance was quite a sensitive indicator of potential glycine deficiency. Now you will see a lot of studies on glycine in literature, and of course they do not always find this effect, but I think it illustrates that if you continue to reduce dietary crude protein, at some stage, under some conditions, you may end up with some deficiencies in non-essential amino acids. So the concluding remarks from this part of the presentation is that low protein diets are very important to reduce emissions. That of course there should be adequate supplementation of essential amino acids, and I really think there is a lot of interesting literature out there studying the effect of the next limiting essential amino acids. And in this way, we will be very successful in minimizing the intake and thereby also the excretion of dietary nitrogen. But when non-essential amino acids may become limiting, you may consider to supply urea nitrogen or ammonia nitrogen as a suitable nitrogen source. But under particular conditions, there may be a specific requirement for non-essential amino acids that may not be covered by your low protein diet. With that, I would like to have a very brief opportunity for questions before I proceed with my next part of the presentation.
Okay, so there is one question. Do we really know the amount of amino acids needed of secondary amino acids, or are we just knowing it at the status for the primary amino acids?
So I'm not really sure. I assume that by primary amino acids you mean essential, and by secondary non-essential amino acids.
I think so.
Okay.
That this is meant.
I think there is not much known about the requirements of non-essential amino acids, and there have been a lot of studies investigating the effects of supplementation of non-essential amino acids under conditions at which they were not necessarily limiting. So I think quantitatively there is not a lot of information out there. And if you look into the effectiveness of supplementation by non-essential amino acids, you find great variation in the responses. So I think the big trick is to find out under what circumstances which one of the non-essential amino acids may become limiting.
Okay.
And in that
Thank
sense,
you.
it may be nice to add that, for example, in poultry, it is known that the one carbon content of glycine may also be very important for the excretion, for the adequate excretion of uric acid. So
Okay.
there are key processes that require non-essential amino acids, but of course you can synthesize them.
Okay. Thank you very much. And here is coming another. What is the optimal ratio of essential amino acids to non-essential amino acids in fattening diets?
Oh, that's a very good and but also very practical question that I would need to study this in order to give an answer. But I think that the report that I showed you from the European Union gives you quite a bit of information that you could use if you really want to compare the ratios of non-essential versus essential ones.
Okay.
I'm very
And
sorry.
maybe one last question. What could be the impact of giving urea by the feed in very low protein level?
Yeah, it could be, and I think also that's the study that I showed, the Canadian study. Is urea will likely be absorbed as ammonia, although in the stomach there may be hydrolysis, and then you also may lose some ammonia. But I think the idea of feeding urea would be that it's absorbed as ammonia and would be effective when non-essential amino acids are limiting. So if the non-essentials are not limiting, it will be excreted in urine. And if the non-essentials are limiting, it may be used in favor of nitrogen retention. The studies that have been performed in Canada, they will show that supplementation in the form of ammonia is more effective than supplementation in the form of urea.
Okay, thank you very much. And maybe one last question, and then we have to go over to the next topic, is can we always say it's always 10% reduction in nitrogen excretion by every percent of reduction of dietary crude protein? Is it a linear response?
Yeah, that's a very nice question. I think it's been shown. There has been a lot of research by Ajinomoto, and that is also incorporated as such in the report that I showed in the presentation. And it shows that within the range of, let's say, 17-18% crude protein back to 12, it's a linear relationship. But I also think that the study from China I showed is that any further reduction to back to 9% crude protein at the end of the fattening period doesn't give as much of a reduction anymore. So I think over a fairly large range it is linear, but in the low range it certainly is not.
Okay, thank you very much, Walter.
All right, I will proceed with the next part of my presentation then.
Yes,
and I will
thank you
focus
very
then
much.
I will then focus on the relationship between low protein diets and behavior. I will show you here some damaging behaviors that are not very nice to see. The belly nosing, if it is, it can be quite gentle, then it's regarded as positive, but mostly is regarded as manipulative behavior and damaging behavior. You see tail biting, I think that's in many parts of the world, ear biting and other manipulative behaviors. Damaging behaviors are a multifactorial problem. There is also here at the site of the European Commission quite a nice practical document on cutting the need for tail docking, and they acknowledge that it's truly multifactorial, and they indicate here the different factors that may play a role. So having adequate enrichment materials, thermal comfort, air quality and light. There is a relationship between health and fitness, And competition is quite important. If there's not enough feeding spaces or not enough space for the pigs, then you may increase the risk of damaging behaviors, but also there is a relationship with the diet, which I will go into a little bit more, and pen structure and cleanliness. And I think under practical conditions, enrichment materials are usually not present to the extent needed to prevent damaging behaviors. Also, competition with high density of pigs in the pen is an issue, which means that we can regard the diet as a risk factor under commercial housing conditions. And we have to go back to, let's say, the early 90s. I think also there was a little bit earlier research in which they found that protein deficiency can trigger the preference for blood. And this was a relatively small scale study, 40 pigs, initial body weight 30 kilos by Fraser in Canada. And they had 4 diets, a negative control diet and a control diet. And the control diet was a negative control plus 17% of soybean meal, which means that the negative control must have been roughly 10 to 12% crude protein. Is difficult to deduce from the paper. And there was a lysine supplemented diet and a diet which is supplemented by a mixture of various amino acids. And the pigs were given a choice to chew on a rope that was either impregnated with pig blood or a clean rope. On the y-axis, you see the mean daily preference score, and this is the difference between the number of times that the pigs were chewing on the impregnated rope minus the number of times they were chewing on the clean rope. And what you see in week 2 and week 4 is that there is a clear difference between the negative control, let's say the low protein diet, and the control diet, which would be a high protein diet. And the authors, this was repeated also in week 4, and the authors concluded that there is a deficiency in certain amino acids that triggers pigs to be attracted to blood, and that could have been was identified as one of the causes of damaging behaviors, in particularly tail and ear biting. But however, this was not always easy to reproduce. This was done in 2002 by McIntyre and Edwards. They looked into different weeks, and they had the same preference score, but I think it was divided by 100, and they did not see a lot of differences between 3 treatments: a control treatment, which was a high protein, high energy intake, a low protein, high energy intake, or a high protein, low energy intake. I'm sorry about this. So no effects in the first 5 weeks, but in the last week, they found an effect in a similar direction. And the conclusion was that the earlier studies in the 80s and 90s were quite difficult to reproduce, although there seemed to be some indications that there would still be some truth in it. And I think it's interesting to note that when you do this kind of study, you focus on the pigs that perform the damaging behavior rather than the other pigs. I also think it's interesting to look at a more recent study that was conducted by one of our PhD students, in which she was using over 500 male pigs in the weight range from 20 to 110 kilograms of body weight, 3-phase feeding, and the study was 2 × 2 × 2 factorial design with a normal or a low protein concentration, and the low protein concentration was 20% lower than the normal. Sanitary conditions was either high or low, and the diets were supplemented or not, 0 supplementation or 20% supplementation, by a mixture of threonine, methionine, and tryptophan. And I'm not going to show you all the results of this study, but I will focus on the effect of the low protein versus the normal protein diet. And on the y-axis here, you see the frequency of different behaviors per pig per hour. And you can see that total oral manipulations of penmates was higher in the low protein diet compared with the normal protein diet. And you see that reflected in most of the individual behaviors. Tail biting was a tendency, ear biting was increased, other manipulative behaviors were increased, toy chewing, total aggression, fighting, and also belly nosing was increased in the low protein diet. And when you look at the interactions with the sanitary conditions, and the sanitary conditions was also a condition that triggered more damaging behaviors, is that supplementing amino acids would affect tail and ear biting in a way that depended on the sanitary status. So it indicates that low protein is a risk factor under commercial circumstances, but it also indicates that these amino acids, threonine, methionine, and tryptophan, are not the complete story because the effectiveness of it depended on sanitary conditions. And it's been known that both the performers and the victims of aggressive behavior or damaging behaviors usually suffer from reduced growth rate. And this is a study by Kamerlingh et al. It's an observational study, so no experimental contrast in a large number of pigs between 8 and 26 weeks of age. The pigs were housed in groups, and they were actually studying the relationships between growth rates and different types of pigs performing different types of behaviors. And unlike what was expected, is that the relationship between growth rate on the y-axis here and oral manipulations was only there for the receivers of oral manipulations, particularly when receiving more than 2% oral manipulations, the growth rate was reduced, and this is excluding clinical cases. There was no relationship between the growth rate and the performance of disturbing behaviors. Another study by Ursula et al. And she measured 480 pigs at different points in time, housed in the groups in a commercial setting, and there was an experimental treatment. It was either the pigs were housed in a barren circumstances or enriched in pens that were enriched with straw. And the conclusions were a little bit difficult to formulate, but what they said is that dependent on the stage of life, tail biters had lower levels of serotonin in the blood compared with neutrals or pigs that were both victims and biters. And to a lesser extent, this was also true for the victims. Now, serotonin, the tryptophan is a precursor of serotonin. So there is an obvious link with tryptophan metabolism there. And I think there has been a lot of research done on tryptophan, but I also think it's obvious now that the tryptophan is not the only amino acid that is involved in the occurrence of this problem. Another thing that I found very interesting in this study is that under barren housing conditions, the tail biters appeared to be more fearful. So aggressive behavior and damaging behavior was coming out of fear. And the concluding remarks would be that damaging behaviors is a multifactorial problem in pig production. Low protein diets are a risk factor, and I think that's particularly true under commercial housing conditions. And it's not a single amino acid that is responsible for this, and mechanisms may differ between victims and performance of damaging behaviors. And which is the end of the second part of my presentation, and I welcome any questions that you might have.
Thank you very much, Walter. So I saw here is a question. We often do see especially with low protein diets, kind of that sows are very agile and not calm. Do you think we could make these cells calmer again by increasing the protein levels again? Or that this has something to do with low crude protein levels?
Possibly, could be. And is this during gestation or during lactation?
What are the questions are coming in the
Okay, dependent. I could imagine that if it's during gestation at relatively low feeding levels, that it may be also a good idea to supplement with a fiber-rich diet, increasing in this way satiety and satiation in these sows. But I could not exclude that the protein level is also a factor that plays a role. I don't have experience with observations in sows.
Okay, thank you. So as far as there are no further questions to this topic, I say go ahead, Bläser, with the next topic.
All right. So my last section is a little bit longer than the previous ones, is about low protein diets in practice. When you look into research conditions, they're usually quite clean when compared with practice. And if you want to find the effectiveness of feed additives or a different dietary strategy, and you hypothesize that it has an effect on health, then usually people have been using, and we have also been using, infection models and sterile infections to test the effectiveness of feed additives or different strategies in pigs. So we've been doing that too, research with coli infection models or salmonella infections, or sometimes sterile infections with for example, with LPS. But I have to say that these are really a clinical setting, and they may be very important on the mechanisms that are involved. But I think it's important to realize that when you do this kind of studies, it gives you it gives you information about the effectiveness under clinical conditions. I want to draw some attention to the work that has been done in different countries. I will focus on our own studies, but I know that people in Canada, but also at INRA in France, they've been doing very nice studies with different sanitary models. And it's there to mimic chronic low-grade activation of the immune system. Now, of course, the immune system needs feeding. So if you activate the immune system, you have an increase in the direct costs for energy metabolism. But there may be also a direct cost for the proteins or specific amino acids that are needed for the immune system. On the other hand, there may be also increased nutritional requirements for covering the associated costs. And looking back into several years of research, I tend to think that these costs may be more important quantitatively than these costs. And I don't have time to go into this topic, but I hope by the end of the presentation, when looking at different effects of sanitary models, you agree with me that the associated cost may be something to focus on. And of course, and that's, I think, the connection also between the different parts in my presentation, is there will be interactions with animal behavior. Also, the behavioral study that I was showing in the previous presentation, the effects were dependent on sanitary conditions under which the pigs were housed. This is a study performed in 2015, and it was with 48 grower pigs of about 45 kilograms, and they were housed in in 6 different farms. They were receiving exactly the same diet, and there were the same pig genetics on these farms. And the complete nitrogen balance study was performed on farm. And what you see for the different farms, you can see here farm 1 through farm 6, and you can see the nitrogen retention efficiency expressed as a percentage of nitrogen intake, and there is a clear farm effect present. So same pigs, same genetics of pigs, same diet, and the range is, let's say, roughly 6% in efficiency. The same study, but a different weight range, 10 different pens of pigs in the same farms, receiving the same diets and having the same pig genetics. Indigestible marker technique was used to measure digestibility. And what you see here in red is the nitrogen digestibility on each of these farms, and in blue, the dry matter digestibility. And if you focus on the nitrogen digestibility, you can see that typically the difference between the highest and the lowest farm would be, let's say, 3 to 4 percentage points in nitrogen digestibility. And I have to say that all these animals were clinically healthy. And I think when you look into these effects of different farms, this has to be related to the housing conditions, the management, and the health status of these pigs. And of course, they are all interrelated. So the management and also the housing conditions can affect the health status of these pigs. And if you want to study effectiveness of different nutritional strategies or feed additives, I think it's important to realize that it would be nice to be able to mimic this kind of variation in health conditions. And that's why we have been working with contrast in sanitary conditions. And I already told you before that there are also other groups in the world doing this research with different sanitary conditions, and they also have received very nice results. You can see the top 3 graphs here, the top 3 pictures here. There are two different experiments with high sanitary conditions, and the bottom 3 are the same experiments, but then the low sanitary controls. They were all housed inside our climate respiration facilities. So all these pens that you see here are actually in our climatized facility. And typically what we do is the high sanitary conditions. We have cleaned pens, disinfected rooms. We daily remove manure, and there's a very strict hygiene protocol. The low sanitary conditions, normal rooms, no removal of manure, no hygiene protocol, and the most important one, I think, is the bi-weekly or weekly, dependent on the experiment, the spreading of manure that is obtained from other farms. And sometimes in some experiments, we have also exposed the pigs to dust. This is a critical thing, and it requires a lot of thinking how you do this. You don't want to introduce an infection here, so you don't want to introduce pathogens. So it's very nice and wise to screen the manure on pathogens before you do this kind of trial. And it's important to realize that you can only use the results of these kind of trials if you do not have clinical outbreaks of disease. A second study that I would like to show you the results of is that low sanitary conditions can increase maintenance energy requirements. It's kind of a large-scale study, 144 male pigs, also in group housing condition, and also 2 treatments. Sanitary conditions, high or low, and a supplemental amino acid mixture, tryptophan, methionine, and threonine, either not supplemented or supplemented by 20%. All pigs received first a week of ad lib feeding and then restricted feeding, and we performed complete energy and nitrogen balances, and we measured the fasting heat as a proxy for maintenance energy requirements. So what you see here on the y-axis is heat production in kilojoules per kilogram metabolic weight per day. And here is the time relative to the last feeding. And the last feeding was here. What you see in the top 2 lines is total heat production. In the bottom 2 lines is activity heat. And the difference between the two is called the resting metabolic rate. And this is usually used as an estimate for maintenance energy requirement. Of course, it's not the same. but it's considered to be a good proxy. And here you can see that the low sanitary conditions have, let's say, 7% higher maintenance energy requirement. So in summary, what this means is that the energy requirements for maintenance are increased by some 7% under low sanitary conditions. And these two are really pictures from this trial. In this trial, Yvonne, who was performing the study, also looked into the utilization of dietary protein. And she was feeding different quantities of protein in these in this trial. And what she found is when you increase protein intake under the high protein conditions, when you increase protein intake, the use of the extra protein is, let's say, roughly 70 to 75%. So if you increase protein intake by 100 grams per day, 70 to 75 grams would be deposited, whereas this was 50 to 55 grams in the low sanitary status. So a reduction of 20% in the marginal efficiency of protein use. And this is kind of difficult to interpret, but it does mean to me that dirty pigs, they need more protein for optimal gain, but they will use it less efficiently. And this gives you the dilemma. If you are feeding a scarce resource like protein, would you rather give it to dirty or to clean pigs? The dirty pigs would increase their performance, but clean pigs would increase their performance more, so you get a better return on your protein investment. So in conclusion, designing low protein diets under practical conditions deserves extra attention. The sanitary condition model is not a disease model, and it's interesting to study relevant differences in health conditions. And for sanitary conditions, digestibility may be reduced by some 5 percentage points, and we've done also different studies, and sometimes it's 6, sometimes it's 4, but it's always a consistent effect on apparent crude protein digestibility. Maintenance energy may be increased by some 7%, and the utilization or the marginal utilization of protein is reduced by 20%. And this is the end of my presentation. I thank you for your attention, and I welcome any questions to any of the parts that I've been presenting.
Thank you very much, Walter, for this excellent presentation. And there is a question, and this is, were the feeders and the waterers kept clean in this experiment?
Yes, we spend a lot of attention on keeping the water supply and the feed supply clean. And I have to say also that training the pigs under dirty conditions to keep the feeder clean is kind of a challenge.
So this is an illustration of the use of the protein in the gut. So you have on the top left side the dietary protein, which is represented, and when it arrives to the small intestine, it will be degraded as amino acids and peptides, which will be absorbed. And this part, which is absorbed in the small intestine, is the digestible protein. And this digestible protein will be used by the animal for maintenance and growth and meat and milk production. Can you move on, please? And thank you. And the second, what is not digested in the first part will arrive to the hindgut. So this is the non-digestible protein. And the problem with this non-digestible protein is that it may be used by the pathogens, which are mostly proteolytic bacteria. So there will be a development of these pathogens. And a second consequence is that the non-digested protein will be also excreted in the environment. So we will also get an environmental impact. Next slide, please. So as I mentioned, we have a solution for this. So that's the Jefo Pro solution. Next slide, please. Yeah, I mentioned this. So it's a fermentation extract, GMO-free, that can be used on all phases of pigs. Next slide, please. And our Jefo Pro solution will help by increasing the digestible protein. And as a consequence, we will have better zootechnical performance. And the second consequence will be that we will get less non-digestible protein arriving to the hindgut. So we will have enhanced intestinal health and also lower environmental impact. So next slide, please. So now we will come to the trials. So first, the German trial. Next slide, please. So as I mentioned, so that's a field trial that we have done. It was done on 1,020 fattening pigs. We started at 30 kilos and we ended at slaughter. We worked on 3 different diets where we had different crude protein levels. So the first diet had a crude protein level of 16%. The second diet had a crude protein level of 14%. And the last diet had a crude protein level of 12.5%. And so we had 2 treatments, a control group with 12 pens, and in a second group where we have added the Jefo Pro solution, and we have added it on top, so no reformulation. And we have measured the performance. Next slide, please. So let's go to the performance. So first, if we have a look at the table, so we have on the second column the control group, on the third column the Jefo Pro group, and on the last column the difference between both groups. And we can see that the starting weight was the same in both groups. But when we look at the end weight, we had an improvement of 1.87 kilos for the Jefo Pro group. And regarding the average daily feed intake, we were also on the same levels. Now, let's have a look at the average daily gain. So in gray, the gray column is the control group, and the pink column is the Jefo Pro group. And you can see that by adding Jefo Pro in the formulas, we managed to improve significantly the average daily gain. And also, interestingly, we can see that we have also an impact on the feed conversion ratio, where we decreased from 2.51 to 2.46. So we can clearly see that we have an improvement in feed efficiency with the Jefo Pro solution, thanks to a protein and amino acids which are more available for the pig. So next slide, please. So we have also made economical calculations. So from the data we have estimated the carcass weight by applying a carcass yield of 76% to the end weight. So this gives us 83.7 kilos of carcass for the control group and 85.1 kilos for the Jefo Pro group. So that's a difference of 1.42 kilos of carcass. We also have made an estimation of the price paid per pig. So we have taken a price of €1.50 per kilo of carcass, and we have multiplied by the carcass weight, and this gives us an €2.14 extra per pig with the Jefo Pro solution. We also have evaluated the total feed cost, so taking into account the quantity eaten per pig, but also the price of the feed. And we had €0.83 extra per pig in the Jefo Pro solution. But when looking at the net gain per pig, so by making the difference between the price paid per pig and the feed cost, we still earn €1.31 per pig. So this shows that even on a low crude protein diet, we manage with the Jefo Pro solution to improve the feed efficiency, and we have a beneficial consequence on the net gain per pig. So next slide, please. So I will present to you a second trial that we have done in Hungary this time. So this is also very fresh results. So we have worked on 60 fattening pigs. We started at 30 kilos and we ended at slaughter. We had 3 different diets. So the first diet was a grower diet, which was provided to the pigs from 30 to 50 kilos of body weight. The second diet was provided to pigs between 50 and 80 kilos of body weight. And the last diet was provided to pigs from 80 to 120 kilos of body weight. And we had 2 treatments, so a control group with the regular formulas, and a second group where we have used the Jefo Pro, but we have made a reformulation in order to lower the feeding costs and formula costs. So what we have done is that on the grower formula, we have made a relative reduction of crude protein of 0.53%. And we have taken out in the formula 1.7% of soya. In the second formula, we have made a relative reduction of crude protein of 0.26% and taken out 1.4% of soya. And in the last formula, we have had a relative reduction of crude protein of 1%, and we have taken out 1.5% of soya. So we have reduced our feeding cost, and we wanted to see what were the results on performance. So let's have a look at next slide. So we didn't have time yet to make the statistical analysis, but here are the raw data. We can see that the initial weight was the same in both groups. The final weight was improved by almost 4 kilos with the Jefo Pro solution. As for the previous trial, we were on the same levels of feed intake in both groups. And we could see an improvement of average daily gain of 49.7 grams per day extra with the Jefo Pro solution. And also we could see an interesting reduction of the feed conversion ratio of 0.09. So in this trial, so we have different things to take as a summary. First of all, we have reformulated, so we have decreased the feeding cost. And in addition, we have even improved the performance in this trial. So what we can clearly see is that the Jefo matrix is safe to be used, and even in a high performance context. So these were the 2 trials, the field trials. And now we will switch on another trial. Next slide, please. Where we have looked at the reduction in NH3 emissions. Next slide, please. So this trial was made in Bangkok University in South Korea. We have worked on 140 pigs of 24 kilos. The trial has lasted during 42 days, and we had 4 treatments. For each treatment, we had 7 pens with 5 pigs. So the first treatment was a standard diet. The second treatment was a standard diet where we have added the Jefo Pro solution on top. The third diet was a low density diet where we have decreased the crude protein by 1%. So we have the standard diet was at 20% of crude protein, and now we came to 19% of crude protein. And we also have decreased of 0.06% the digestible lysine. And the last treatment was the low density diet with the Jefo Pro solution added on top. And we have measured, so of course, the performance, but also the gas emission in this trial. So let's have a look at the results now. So first, to explain you, the gray and the black bars correspond to the standard diet and the low density diet. And then the bars which are in pink correspond to the treatments where we have added the Jefo Pro on top. So when we have a look first at the average daily gain, the first thing we can see is that we have a reduction of the average daily gain when we compare the standard diet to the low density diet. And the second thing that we can observe is that we have an improvement of the average daily gain between when we add the Jefo Pro solution. And this is available. This is true for the standard diet, but also for the low density diet. And when we have a look at the gain to feed ratio, we can see, so the first thing that we can see is that with a low density diet, we reduce this gain to feed ratio. And this difference is significant between the standard diet and the low density diet. And when we have a look at the results with the Jefo Pro solution, we can see that we have a significant improvement of this gain to feed ratio. So we can see that we have first improved the performance and the feed efficiency in this trial. And this is true for standard diet and low density diet. So now let's have a look to the NH3 emissions. So the first thing we can see, and I would like to make you a remark, is that when we compare the standard diet and the low density diet, we are on the same levels of NH3 emissions. So this can be explained by the fact that when we reduce the density of the diet, perhaps the ratios of essential amino acids have not been taken into account, and also we keep on the same level and proportion of non-digestible protein. And the second thing I would like to show you is that with the Jefo Pro solution, added on top of the standard diet, we can see a reduction of the NH3 emission, and we can observe the same thing also on the low density diet. So in this trial really illustrates that we can significantly improve the environmental impact of the diet while improving performance and feed efficiency. Next slide, please. So as a conclusion to this presentation, we have seen that the protein in animal nutrition is important in terms of availability, of cost. We can see it clearly right now, and also as environmental impact. And our Jefo Pro solution enables to have a better protein digestibility by making the amino acids more available for the pig to use for their own needs and growth. And we have also less non-digestible protein arriving to the hindgut, which has, as a consequence, a less production of pathogens, as there is less nutrients available for the pathogens, and also better intestinal health. And the Jefo Pro helps in having a better feed efficiency. This we have seen over the different trials I have presented to you. And we have a globally beneficial impact on the environmental with the Jefo Pro solution. So thank you very much. I'm really sorry for this technical issue, but if you have any questions, I would be really happy to answer you.
So thank you very much, Anaïs. I think it was a very heavy topic to do this presentation without seeing anything. So thank you very much. And also thank you very much to Walter for being so spontaneous to take over the presentation of Anaïs, at least at presenting it or showing the slides. But now we are coming over to the questions. I received several questions, but due to the fact that the time has already is already that far, I'm going to ask you the question that I have received in the meanwhile. It's a very simple question. Is the Jefo Pro solution also usable in other species, or is it just adequate in swine?
No, it's very interesting because the Jefo Pro solution can be used also in poultry. We have very nice results also to show in poultry, and also concerning the sanitary effect and gut integrity. And we also have a solution, the Jefo aqua solution, which can be used in aquaculture, where also the environmental questions are really important.
Mm-hmm. And oops, now I closed my screen. So this includes also not only fatteners, but also piglets and sows and any. Okay.
Yes,
okay.
Exactly.
And the next question is, if this happens just on protein sources based on soy, or is this solution also efficient with other protein sources?
So that's a very good question. Thank you, Ursula. So the Jefo Pro solution works on all kinds of protein sources. We have some different in vitro trials where we have measured the improvement of digestibility, and we can see a clear improvement with other protein sources such as wheat DDGS, sunflower cake. So it works really on all type of protein, and we also see similar results in aquaculture, so where we have also different other types of protein raw materials.
Mm-hmm. And here's a very interesting question. I think it can be given to both of you. Do you think that the Jefo Pro solution can also have an impact on the behavior of the animals in low crude protein diets?
Walter, do you have any?
Well, potentially yes. If it improves the absorption of amino acids from the intestinal tract and thereby reduces protein fermentation end products, it could lead to more amino acids being absorbed, and in that sense, it would be the same effect as changing your protein concentration in the diet. There are also possibilities that impaired health because of excessive protein fermentation can have an impairment of behaviors, but that I don't know. I mean, I can imagine that it's there, but I don't know of the evidence.
Mm-hmm. And I would like to add something on what you have said, Walter. We also think that there can be an effect on feeding behavior, by the sense that when you have a more regular digestion of the protein, you will also have a more regular ingestion. And we have some results in sows which go in that sense.
Mm. Okay, so thank you very much. I think now we are already over the time we have postponed at the beginning, but I don't care. So it's not a problem because we had some technical problems, and I think we made it really good in the solution, like a team, that everybody tries to support us with giving access to computers, like Walter did. So I say thank you very much for your support, especially Walter, and of course also for you, Anaïs. And if there are any questions which could not be answered during this meeting, we are going to write an email or anything like that. And we are also asking then again the support of Walter for the questions. So thank you very much. Enjoy the afternoon, and we will stay in touch. Au revoir.
Thank you very much. Bye-bye.